Okay, so many of us here have these big libraries of birdsong, and we want to be able to use that song to learn some things. For example, about sequential structure of our song, things about syllables, categories, features that exist in our song, like how attractive a song is, as well as age and sex, things like that, and the parallels that exist, or the relationships between physical space, neural representations, and perceptual representations of our song. So to help ask these questions, I developed a package which I'm calling Animal Vocalization Generative Network, or AVGN. And basically this is a one-slide abstract of what AVGN does. It allows you to take your library of song and project that song into a low-dimensional sort of syllable space or song space. It allows you to take that song and sample from that low-dimensional song space and generate novel, kind of smoothly varying sets of stimuli that you can use for your experiments. It allows you to cluster in that space and automatically label your song without having to do any sort of hand labeling, so it's in an unsupervised manner. And it allows you to do things like take those trajectories in that low-dimensional syllable manifold and learn models like this first-order Markov model here. So this is a series of tools that are basically centered around two things: dimensionality reduction and generativity. We're on the dimensionality reduction side. We're basically taking our library of song, converting it to a library of syllables, and then projecting into that latent representation, or that birdsong sort of space. And the generativity is the opposite. We sample from that latent representation, that low-dimensional space, generate syllables of kind of smoothly varying stimuli that we can use for different sorts of playback studies or neural analyses, things like that. So the key part of this is that we want to learn a low-dimensional latent representation of our birdsong, or in other words, kind of a space of our birdsong, where each one of these little black dots that you see is a single syllable, here of Cassin's video song, so one of around 10,000 syllables, I think, that you're seeing here. And what we want to be able to do again is both translate from syllable space into this latent space using dimensionality reduction, and also be able to translate back from that latent space into syllable space using generativity. So basically there's two parts to this short talk. The first is how does it work, and the second is what can I use it for. I'm going to be showing examples from two data sets of birdsong that were freely available on the internet. First, Cassin's video and Bengalese finch. So basically everything that you're going to see today is in a Jupyter notebook on this repository, which you can go online, and there's even links for how you download these data sets, so you should be able to run everything yourself. Some of these might require a GPU in your computer, but there's Google has GPUs available for you to use. You can, even with a bad computer, you can go online and use these. So that said, this is all species independent, so this is a few of the species that I've used. So it's not just birds. This also works with marmosets. I've played around with humpback whales and different types of bats. And hopefully I'll be able to put the rest of these examples on the internet soon. So first, here's an overview of how this works. What we're doing is we're taking our library of waveforms of vocalizations, we're segmenting them, we're pulling out the bouts, and we're segmenting into syllables. This is actually the part that I'm going to talk about the least, and it's kind of the part of my presentation, or this package that I like the least, and I think can be improved a lot upon by other sorts of algorithms that exist. I know Yarden's going to be talking about supervised segmentation, and I think that a lot of these ideas can be pulled together. But so we take these, we kind of create our library of syllables, and then we have these number of options that we have available for kind of specific use cases. So we have these 3 autoencoders on the left, and I'll talk about what those are, and a dimensionality reduction UMAP on the right. And basically, from left to right, these different models are better for different things. So the more left models are better for things like generative modeling, and the more right models are better for things like clustering. So basically, this is the different sorts of latent representation that are produced. So the variational autoencoder just produces a Gaussian latent representation. UMAP produces something that's very clusterable, and the multidimensional scaling autoencoder is somewhere in between. The Gaia autoencoder that I'm going to talk about a little bit less or not at all is producing a different form of latent representation entirely. And again, each of these different models are good for different things. So UMAP is good for clustering, multidimensional scaling is good for clustering and visualizing, whereas these kind of two to the left are better for generative modeling, so producing nice little syllable spaces. So I'm going to focus on the autoencoder side of this, because they do both of the kind of primary components of what I'm talking about. They do generativity and dimensionality reduction. So those 3 types of models are called a form of neural network network called an autoencoder. This is a deep convolutional autoencoder specifically. So basically the way that this works is the left half is the encoder and the right half is the decoder. The input and the output are both the same syllable. So basically you're trying to reproduce your output as your input using this neural network. As you go through, each of these layers, which are the green boxes, are interested in a specific region each of the layers contains a number of neurons that are interested in a specific region of your spectrogram. And as you go deeper into the network, the kind of pieces of the spectrogram that they're interested in become bigger and more complex. So they learn kind of receptive field type things. As you get into the middle of the network, you get this latent layer where the activation of these different neurons corresponds to those low-dimensional kind of components of your song, so the x and y axes of the scatter plot that I showed you. And then the decoder does the exact opposite thing. It inverts from that latent representation back into the spectrogram. So to reiterate, the encoder does dimensionality reduction. So we go from our library of syllables to our latent scatter plot of syllables by projecting each one of these syllables in through the encoder. And the decoder is generative. So basically what it's doing is I'm uniformly sampling from this latent syllable space, and I can project through the decoder and get this kind of grid of continuously sampling syllables. And these are syllables that the bird never actually produced necessarily, but they're syllables that kind of fit inside that space.
What's the dimensionality of the latent layer?
It depends on the type of network that you're using. So some of the variational autoencoders will go up to 64 dimensions, okay, whereas for these visualizations I'm just using 2 dimensions because it's easy for people to understand.
Okay.
So one of the first things that you notice about these latent representations is that they form clusters. So here we're looking at some Cassin's video song, and you can kind of see these nice little black clusters that exist. So what we can do is we can use automated clustering techniques like here HDB scan, and we can compare the clustering that exists that we did automatically with hand labels, and what we see is on the left, these are just a couple of measures of similarity between labeling schemes, so the hand labels and the HDB scan labels. And what you see is that they're very similar, basically. And if we do the same thing with UMAP, which was again the more clusterable latent space, we get an even better kind of similarity. So the similarity between the UMAP and the HDB scan labels, or the automated, the similarity between the hand labels and the automated labels are very high. So you can see if I go back and forth that there's a lot of overlap between these two things. In this example, there's this one cluster here that really stands out to me that is a hand label that kind of has 2 overlapping clusters, whereas the automated technique calls it the same thing. And I think that actually points out something interesting about the difference between hand labels and automated labels is that the automatic labeling is going to inherently be kind of a principled manner of segmenting your syllables out, whereas hand labeling, we're prone to human biases and errors. Okay, so here's an example of several Bengalese finches, which we're using this UMAP technique on. We project them into this sort of syllable space. We cluster them into different categories, and then we can do things like plot the kind of trajectory of song in that sort of syllable space, or that low-dimensional space. And then we can, do things with that transit. So here you're seeing things about the transitional information. Like I said before, we could do things like learn Markov models of this song, stuff like that. And we can also take these labels and visualize them on our spectrogram. So again, this is completely unsupervised. I know nothing about these birds. I didn't look at these bird songs before. But what we can do is we can do these kind of classic plots that you've probably all seen before of kind of color plotted over top of the syllables. And you see that there's a lot of well, if you take a deeper look, you can see that there's a lot of uniformity in the labeling of the syllables. So this is just each one of these rows here is represented by a column here. So you can see the patterns that kind of exist in these probably about 200 Bengalese finch songs that were entirely unlabeled. Okay, so the more interesting part about this to me is that it's actually the generativity side. So basically the way that works again is that we take our latent space, we sample from it, we push it through the decoder, we get continuously sampled syllables, and then we can reconstruct waveforms from that. So this is an example of 3 different reconstructions. So here's from the multi-dimensional scaling autoencoder, so the one that's better at doing clustering and so is kind of therefore less smooth, so there's going to be stronger boundaries. The variational autoencoder, where we have kind of a more smooth latent space, and then a variational autoencoder on European starlings. So this is an actual example that I used in an experiment that I actually have a poster for today. So just play these for you, but I didn't actually set up a a microphone. So you saw kind of a quick transition in that. This should be a little bit of a smoother transition. And as you increase the dimensionality of the variational autoencoder, the transition will become more smooth. This is a European styling song, so this is an example from an experiment that I'm currently doing. So again, that was just a line drawn in that latent space between two syllables, and we're just kind of transitioning on the line, recreating syllables from that interpolation. So I have an experiment, a poster about that. You can come if you want to hear more about either this method or that experiment specifically. Come out to the poster. But this is all work that I did with my advisor Tim Gettner and Marvin Tilk, who's in my lab and who's also here, worked on this with me. So thanks for listening.